Hello everyone, my name is Abby and I'm going to be walking you through Yeast ORFan Gene Project Lab Module 4. In this module, we will be using TEACOFFEE to observe the conservation of amino acid sequences through multiple sequence alignments based off of the related alignments found in BLAST. We're then going to create a sequence logo of this data using WebLogo. So let's get started. I'm now going to go to the Yeast ORFan Project webpage and open up the worksheet and open up the guide. And on the guide, the first page here, you'll see that there's an amino acid chart that you'll want to reference later. So what we're going to be first working with is TEACOFFEE, and we want to start by going to the SGD under the sequence section. Under the protein section, and we're going to do a BLAST at NCBI, which is under the resource section. And we're going to do the BLAST, and we don't have to change anything, just click BLAST and go ahead. And then wait. Alright, and once your results have loaded, you are going to want to click the Select All button, uncheck that so everything is unselected, and you're going to go through and select 10 to 30 different organisms that are not Saccharomyces cerevisiae and have an appropriate E-value. So you're going to want to go through and do that. So you don't always want an E-value super close to 0 because this can produce misleading results since these may have functionally diverged from other homologs. So in this case, some amino acid residues that are actually important for function in your protein may not appear to be well conserved in the sequence alignment, so you're going to want to sample a wide variety of E-values. And I'm still using MEC1, and all of mine have an E-value of 0, so it doesn't really matter which ones that I take for my sample. Alright, and once you have like 10 to 30 of these checked off, you'll want to scroll to the top, and under the download section, click the FASTA complete sequence. And that should open you up to a seq dump text file, and you're going to want to save that onto your computer for what we're going to do next. So we're now going to work with the TEACOFFEE server, and I'm opening up the website from the guide. And I'm going to upload my file. And uploading my file. And then everything should be all set, so I can click submit and wait for it to load. And it's going to, at least for me, it loads at the top. And then it's going to take you to this page for you to wait for it to load. And this is what it'll look like when everything's loaded. And I'm going to click Show Color, so that's going to color code everything by amino acid property and similarity, so it's easier to look at and visualize. And that takes a little while to load as well. Alright, there we go. And this program has the same annotations as the other programs that we've used, where the asterisks at the bottom would mean everything is completely conserved, so they all have methionine going along it, and then the colon would mean that it's conserved in terms of all of the amino acids have similar properties. Like they're all nonpolar, for example. So you can look through that, and what we're going to want to do is we're going to make notes about what sections are highly conserved, so which ones have a lot of the asterisks and the colons, and I'm going to do that by taking a screenshot of it. And you can do it whatever way you want. You can take a screenshot, you can just comment about one thing or another. That's honestly up to you how you want to comment on it. And I'm going to find another pretty highly conserved region to take a screenshot of. And we'll do that one. There's a lot of asterisks. And place that in as well. Alright, so now I think I'm all set to work on the WebLogo. Alright, so I'm going to go and download this. And open the WebLogo page. And this is going to help us visualize our results from the TEACOFFEE alignment even better. And this is how mine downloads, so I just am going to copy and paste everything. If it downloads for you as like a text file doc, great, you can upload that to the WebLogo server. But for me, this is how it appears, so I'm just going to copy and paste it. And I'm going to paste it in there. And then under the advanced logo options, under the multi-line logo, I'm going to check that box. And then I am going to submit it. And this is what it first appears like when you create it, and you have to zoom in and out with your mouse. So this should look similar to something, to one of the other programs we used before. And for this, again, we're going to be looking for regions that are really conserved, so we're going to want ones with the really big letters, not a bunch of small letters. So I'm going to take a screenshot of this section because we have this one section that looks to be all one type of big letter. So that's fairly conserved. So we have this like F, phenylalanine, and the number below, it's amino acid 941. It's the only letter there, so it is the only amino acid appearing at that position for all of the different species in the alignment that we compared it to our original amino acid sequence. So this just basically gives it a better visualization for you to decide whether or not you think it's worth looking at. And then for this one, oops. And then for another example, we have this L for leucine number 942. And if you look, you can see another amino acid under it, like that little green section. I'm not sure what amino acid it is, though. I can't really make out what it is, but a bunch of them just have a bunch of little ones squished under a big one, or a bunch of small ones squished together, so those ones would not be conserved. So again, I'm going to take screenshots and just paste them in here under the logo notes. You could comment something else if you did. Again, if you didn't want to screenshot it, that's fine. It's up to you, or what your professor wants you to do. And then this is asking if there's anything new that you can see. So then what you're going to want to do is compare whether or not you missed something and looking at the WebLogo versus the TEACOFFEE alignment, and take it from there. And in the guide, there is a chart for the amino acids, and they're color-coded, I believe, to both of these websites. So you can take more time to look at what different properties of amino acids are conserved throughout your gene and the other genes that we're comparing them to in the alignment. And as you'll see right here at the page I'm scrolling to, there's a section that's gene context. And that's no longer a part of the SGD, so if you have an outdated version of this guide, that'll show up as well. Thanks for watching this video, and I hope that it was helpful. For more information, go to theyeastorphanproject.com, And this video was funded in part by the National Science Foundation. Thanks!